Hey, welcome. So today we're going to be working on a chromatography lab. In general, chromatography is a wide variety of different lab techniques that is used to separate substances into their individual parts. So this can be used to figure out what makes up one substance. It could be used to help figure out an unknown substance by separating it into its individual parts and comparing it to things that we know already. So the simplest type of chromatography, and the one that we've already done in class, paper chromatography, typically is used to separate inks. So for instance, if I had 3 known inks, I could separate them into their components, and then I could compare an unknown sample to the known samples to help figure out where my unknown came from, as well as what it's made of. The ones that we're going to be doing for the rest of our chromatography labs, we're going to be doing 3. They're more complex and they're known as column chromatography. And the techniques that we are going to do are going to either separate molecules based on their size, their charge, or their hydrophobicness or hydrophilicness, so how much they like or dislike water. So we're going to go through those 3. Today we're going to focus on gel chromatography, and we'll have a scenario we're trying to solve here. So typically chromatography consists of separating molecules based on various characteristics. So the one that we're going to do today is based on size. And the way that it works is by having molecules have to choose between a mobile liquid phase or a solid phase, which is stationary. So in our paper chromatography lab, our stationary or immobile phase was the paper, the matrix in the paper, whereas our mobile or liquid phase was the liquid running along the paper. Today we're going to do gel filtration chromatography, and this is what our matrix will look like. So here our solid phase is in the form of beads. We're going to talk about these here in a minute. Beads, which are here in this more jelly-like substance here at the bottom. And then our mobile phase will be the liquid that we filter through this and separate into its components. So before we actually start the actual lab, I want to read you the scenario that we're trying to solve. So this is an example of a real-world situation that could be used solving chromatography techniques. So let's read it to you and you can follow along on your end. So the Rye River, which flows near your school, has large numbers of dead fish that are suddenly seen floating to the surface, and we're trying to figure out what's killing these fish. So the things that we know, the floating fish are primarily bottom-dwelling fish like catfish and darters, as well as the fish who feed on the bottom-dwelling fish like sunfish and minnows. All the fish that we see are dead. They all appear to have skin lesions, reddening or inflammation of the fins, as well as raised scales. And we believe that since all of them have common symptoms, there's probably a common cause for the fish being dead. So we can conduct a quick search on fish kills on the internet, and we figure out that all the symptoms that we're seeing are similar to those caused by rufinotoxin, and we're going to abbreviate that RT. So I may say rufinotoxin, or I may say the toxin, or I may say RT, but I'm referring to the rufinotoxin, which we believe may be killing these fish. So here are some things that we find out about rufinotoxin in our internet search. So we figure out that rufinotoxin is a protein that has been used in many fish kills previously. It is known to bind to heavy metals, therefore it tends to localize to the bottom of the waterways. The toxin has a red color, and it has a molecular weight of 13 kilodaltons. For some of our questions, that's going to be important, 13 kilodaltons. And its isoelectric point is 7. An isoelectric point of 7 means that if the toxin is found in an acidic environment, the overall charge is going to be positive, whereas if it is found in a more basic environment, it's going to have a negative charge. So that specific information about the charge, that will not be important for this lab, but we'll want to remember that for our lab 2 when we're separating based on charge. So there are sources. We're looking for sources of possible ruffinotoxin near the Rio River, which could kill the fish. And in our search, we find that Acme Widget Company is located upstream from where the dead fish have been found. Through further investigation, we find that ruffinotoxin is a byproduct of the manufacturing of their widgets. But Acme Widgets categorically denies that ruffinotoxin is in its effluent and says that only harmless chemicals remain after they process the waste. As proof, they point to the color of the effluent, which is greenish brown with no red color. Remember, ruffinotoxin was red, and they said it's not red, therefore there's no ruffinotoxin present. The definition of the word effluent is going to be a liquid industrial waste. So here is our sample that we have collected of our waste from the Acme Widget Company, and our goal here today is to figure out if ruffinotoxin is present in this waste. So there are a few questions to answer before we actually get started. So go ahead and pause, and this will be on page 2 of your lab, and answer these questions. So question 1 asks, can there be ruffinotoxin, which is a red chemical, in this effluent, even though it looks greenish brown? So again, this is greenish brown. It's not red. I know it's hard to see here, but it is a greenish brown. Could there actually be ruffinotoxin, which is red, in here? Question 2, if ruffinotoxin could be there, what are some ways that we could use to separate the components of the effluent? So think back to what we read about, what we know about the ruffinotoxin, and how could we separate these components? Summarize what we know about the ruffinotoxin for question 3. Question 4, how can we use the properties of the ruffinotoxin to determine if it is present in this substance? So based on what you know, how could we figure out if it's in here? So go ahead and answer those questions briefly, and then, so pause, and then, okay. So, some things that we know. We know ruffinotoxin. We said, if I flip back here, we know it had a molecular weight of 13 kilodaltons, which is relatively small. So the first thing that we're going to do is try to separate these components by size to see what's in here. So to do this, first I'm going to do some lab safety because the chemicals we're working with here, and I'm going to turn the screen so that you all can see what I do. Now I will say, there may be a few points in here if we wait. We might have to do some waiting, but I'm planning on talking during those to give you some background information. So make sure you're watching as well as listening. So this is my weird little setup that I have here with my column. So step number 1 says, remove the stopper from the top of the column. And place a beaker underneath it. So first I'm trying to, I might should have pulled that top off before I hooked it up there. There we go. Alright, so take this off, and I'm going to have my little beaker underneath, remove the end cap, and allow it to drain. So this is going to be dripping slowly. So the way that gel filtration chromatography works, in order to keep the gel preserved so that it works when we're actually trying to filter our toxin, we have to keep the matrix inside moist or wet. So first, we're letting our liquid flow through. So we want it to flow all the way through until it stops dripping. And what this is going to do, it's cleansing out the column to make sure that we're not going to have any contamination when we actually put our sample inside. So again, I'm going to go do some a little background information on gel filtration chromatography. So gel filtration chromatography separates molecules based on size. So the ways that it works is that within this column here, there are small little beads. They're very hard to see. It almost looks like a gel, but there are small beads in here that have they look similar to like a wiffle ball in PE with like lots of little holes in it. So when we put our unknown sample in here, the larger molecules are going to navigate around the beads because they can't fit in the small holes that the beads have. But the smaller items are going to be navigating through the holes in the beads from one bead to another. And what that's going to lead to is we're going to have the larger molecules going through more fast, more quickly, because they're not navigating through the beads. So since they're not navigating through the beads, the large molecules come out first. So the largest component in our unknown here is going to be the first thing to come out, whereas the smallest is going to be the last to come out. Alright, so that was step 1. It stopped dripping now. So step 2, add a fluent sample to the top of the column bed using a transfer pipette. So I am going to add in some of our toxin here. Alright, it's nasty looking. Get my little sample. 0.3. Alright, so I'm going to add this into the top. And the thing about this is, for this lab, we have to pay really close attention. Because what's going to happen, typical types of chromatography may require further processing of the components as we collect them. The cool thing about gel filtration chromatography is as it separates, they're going to separate by color. So all I have to do is look out for the individual colors and collect them. And I bought this special little vial here. I'm going to use this one to collect anything that might look red. And I have some other test tubes here to collect any other bits that we see. So we want to pay attention and be very careful and ensure that we are looking for colors as they come out. So step 3 says, when the column has stopped dripping, add a few drops of loading buffer. Allow it to enter the column. When it stops dripping again, add some more. So I'm going to add some more in here. I'm going to go ahead and squirt some in the top. And as this happens, again, we want to be looking out for the different colors so that we can collect the different colors. And if you look closely here, we can already see some color separation happening here. So I'm seeing some, this is like a, still kind of dark, but I can kind of see some blue starting, but at the top I also see some red and pink starting to form. So we want to watch carefully, and our goal is that when the first color makes its way to the bottom, I will collect the first color drop in a test tube, and I want to keep the colors as separate as I can. And the way that I can control what's coming out when is how quickly I put in liquid at the top. So I can kind of control the rate at which our liquid is moving through by how quickly I put things in at the top. So now that our colors have made it down here, I'm going to add at the top a bit more slowly, just because I want to be sure that I am in control of collecting my colors as they come out. If I was to fill this all the way up, it would happen so quickly, it would be hard to keep up. Alright, so I see it starting to turn blue, so I'm going to switch over to this test tube here, and I'm going to use this test tube to collect my blue color. And my goal is to collect only blue here. So as soon as I see the blue stopping, I want to quickly transfer over. And as we can see, this is going to be quite a quick shift here. So I might have a few more drops of blue here, but if you can tell there at the bottom, it's starting to transition to pink. So I'm getting really close to pink here, and pink is kind of close to the red I'm looking for, so I think I'm going to get out my tube here. So if you see here, there are two colors left. There's the pink here at the bottom, and now a blue at the top. Let's see, I don't know how well you all can see that. Let's see if I can back it here. So I have pink, and I have some yellow at the top. So I want to collect those in two separate vials because the pink might be my toxin. I'm going to collect it in this little special one because we're going to have to further test this to be sure if it's our toxin or not. So I'm going to keep adding here. I'm collecting here. My drips are pinkish in nature now. And again, I want to do my best to collect these separately, so I'm adding drop per drop at the top. I know it's hard to see, but I'm only adding one drop at a time in the top. That way I'm carefully controlling what's coming out at the bottom. So again, gel filtration chromatography separates molecules based on their size. The largest substances come out first. So the blue that I have collected, that would be the largest because it came out first. This pink would be middle in size because it's coming out second. And the yellow, liquid at the top. The yellow would be the smallest because it's coming out last. And again, our substances are slowly working their way through the beaded matrix. So what I'm seeing here is still pink. Oh, nope. So it's starting to turn yellow, so I'm going to quickly switch over there. So here is our pink liquid, and I'll show you all of them side by side here in a few minutes. So that was our second color. Now our third color here is that very light yellow. You might be able to see. And now I only have that one left. So since I only have one color left, I'm not terribly worried about going too fast because I only have one substance left. My goal would have been not to cross-contaminate. So it's still dripping here. See if I can back it. There's still some yellow in there, but it's so light it's hard to see. So I still have some yellow there at the bottom. And I was initially worried that this weird little setup that I have here wouldn't have worked that well because I don't have my actual lab equipment, but it's not going too bad. So I got a little bit more left there at the bottom, but for the most part, most of it's out. So I'm gonna let this finish processing through. And in order, so we're almost done with yellow band. As soon as all my yellow is done, I'm not gonna just fasten it up and call it a day because I want to be sure that my column is fully cleaned out. So I'm going to put this waste beaker back underneath. And I am going to fill the top back up so that I'm essentially rinsing out this column for the next person who uses it. And again, because gel filtrations, they need to stay wet. After this goes through, I need to be sure to fill it back up because they should never dry out. But I'm going to split this clean out to the side here. Turn you back around to face me. And I'll show you what we collected using this white background. So, again, gel filtration chromatography, what I was doing was separating our unknown waste product from the Acme plant, and our goal is to figure out if the ruffinotoxin is present here. So what we did is we used gel filtration chromatography to separate the components of this by their molecular size. And when I did that, we got 3 different components. The first one that came out was a blue component. Because this one came out first, it is the largest. So here is our blue. The second one that I collected was a pink color. Because this came out second, it is the middle as far as molecular size. And the third that I collected was a yellow color. Now the reasoning behind putting this one in a special little vial is because when I saw it come out, my mind said, oh, ruffinotoxin is red. This looks like it might have something in it that could be red. Let me collect that because we're going to want to further analyze it. So we have no confirmation that this pink one yet is a ruffinotoxin. We know that ruffinotoxin is red, but this isn't quite red. It's almost like a brownish, pinkish color. So it's not, if you look at it, it's not really pink. It's almost like a dirty brown color. So it's not red, but we want to further analyze this just to see what could be in here. And that'll be our part 2 of our lab. But first, your lab, here's your lab book. There are some analysis questions that go with this gel filtration chromatography portion. So first is, what are the colors that we collected? So what color samples did we collect? Question 2, does this mean that there is no ruffinotoxin in the effluent from the company? So because we didn't collect red, does that mean it's not there? Question 3, if the ruffinotoxin is present, which sample collected by gel filtration is it likely to be in? 4, can gel filtration separate rafinotoxins from all proteins? So for that one, I'm going to beg you to think, what if we said that rafinotoxin has a molecular weight of 13 kilodaltons? What if there was something in the mixture that was 14 kilodaltons? Would it be able to separate those as easily as these ones separated? So think about that. Question 5, will gel filtration separate rafinotoxin from a protein that is 15 kilodaltons? So think about that. And the last question, what other characteristics of rafinotoxin can we use to separate it from other proteins that are close to size? So what these questions are essentially saying is, this is a brown liquid. Whoop, there we are. This is a brownish colored liquid. There could be multiple things in here. Because rafinotoxin is 13 kilodaltons, there could be something that's 13 kilodaltons in here. The rafinotoxin could be in here, but it could also be mixed with something else that we couldn't separate very easily. So what other properties does it have that we could utilize to help separate it out? So again, make sure you answer your lab questions. They're going to be embedded in the video. And I will see you for part 2, where we analyze our little brownish pinkish liquid.